my presentation is slightly based on what Mr. Sohail just gave. It's an extension of the same. Actually, giving a brief introduction of our company, we are totally into cleanroom partitions, turnkey solutions for HVAC also. And let's see. Now, a simple question to a layman is what is a clean room? It's not a open room. It doesn't mean a clean room, मतलब वो एक open room हो गया. It's more like a total temperature controlled environment, controlled humidity, dust control, contamination control. These controls help the pharmaceutical companies to maintain the sterile, safe, and GMP compliant manufacturing environment. And the clean room is classified into non-classified and classified, in which in a non-classified area like warehouse, then raw material stores, then the passages, you can have a normal ventilation system, whereas in a clean room you need to have a HVAC with HEPA filtration. The sources of clean room contamination personal के अंदर भी even a person sitting or walking can generate dust particle, which is not actually allowed in a pharmaceutical. Now that in the main today's talk is based focused on three critical areas. While designing a clean room, why it fails in audit. Even if a system is designed in such a way that HEPA filters hai, the flow is there, but then also it fails. The reason being that everything is right. जब हम देखेंगे everything will look right. The system is breaking but not the filter. So you have to fix the system, not the total symptom. Or the airflow calculation है. So the design होता है that critical aspect while designing of a clean room or HVAC zoning, room air pressure gradient and air lock, which is designed as per grade A, grade B, grade C, grade D, then maintaining the air changes per hour. If you maintain this, there's no chance for failure in a USFDA or any GMP audit. These are the different classes. Grade A, grade B, grade C, grade D as per EU GMP norms. As per ISO, it was 5678. And as per federal standard, 100,000, 10,000, 1 lakh. This was the old standard. Now, in the HVAC zoning, the rooms and the layout have to be, the rooms have to be identified like in sterile areas, the non-sterile areas, potent drugs, then these are all the grade A, grade B, grade C, grade D, all have to be designed accordingly, and a single AHU has to be given for each and every unit. Both grade A and grade B can have a single AHU, but the grade C, grade D have to have a totally different AHU for it. Other this will prevent total cross contamination. Hello, Sohail sir, this is for your extension only. Now, grade A, aseptic area, grade B, grade C, grade D, grade E. All these have to be taken into account while designing the clean room. Considering the activity in each room, the pressure level is determined. So, the air leaking from the door always moves from a clean to a less clean space. The airlock rooms are introduced to help maintain the pressure differential. This is most important. A barrier has to be given in between the two areas. The minimum pressure difference between the rooms or the difference between them must be 15 Pascal, and same classes it can be 7.5 Pascal. Airlock schemes may be cascade, bubble, or sink type. The most common is the cascade type. This is like the grade A, grade B, grade C, grade D. All will be having 4 different AHUs for all the 4 different areas. And in between the two areas, there's a separation has to be given, which is called airlock concept. The cascade type, as I was saying. This is the cascade type from high pressure to medium pressure to low pressure. This is how it has to be designed. Like this. Then air change per hour is the third thing which has to be taken into account. So CFM has to be decided, the HEPA filter CFM has to be decided as per this only. This is a total calculation. Then comes like UC labs are given on the first floor and not on the ground floor. There is a contamination control होता है उससे the ground है 10 microbial first floor पे it's least. Then vibration control to the instruments नहीं होंगे. Separation from production area, controlled access, environmental control. Then green room validation has to be done DQ, IQ, OQ, PQ. Then at the end we can say what is clean room design purpose. Then why clean rooms are designed at 20 to 25 degrees centigrade temperature and 50 to 55 RH. This is a thumb rule. It is basically for an operator comfort. There's one operator who is wearing 3 layers of ground mask, hood, glove. If we add 28 degrees centigrade and 70% RH, then you would have created a walking sauna होगा उनका तो it would have total he would not be able to sit there. The microbiological control. The microbes love the warm and humid weather. About 60% RH modes like asparagine feel they are on vacation in Goa. So keeping the RH to 50 to 55 make sure your clean room is a complete resort. And static control. Too dry hoga, the static electricity will be there. So always the particle sticking will be there everywhere. Like the capsules sticking to machines. Then regulatory, as per regulatory norms, EU GMP Annex 1 and WHO guideline, they don't bring 20 to 25 and 50, 55 RH as low. But it's the safe zone for all pharma industry. The bottom line is above set temperature and humidity just isn't comfort, it's science. Compliance and survival in pharma. So if you tell somebody कि why 18 degree centigrade क्यों नहीं रखते 60% RH क्यों नहीं रखते you can tell them कि we are not running a spa, we are running a clean room. For that reason, this has to be, these are the strict norms to be followed while designing a clean room. Thank you.